and Computer Engineering in University of California, Santa Cruz. He is also a co-founder of Kaito Tech Medical and Cruise Foam. He received his PhD in applied physics from Stanford University, working on novel nanofabrication strategies and carbon nanotube devices. His research focuses on bioelectronic systems, materials, and devices for biological control in regenerative medicine, synthetic biology, and their translational applications. His work on bioelectronic transistors and shock electrosensors has been highlighted in the New York Times, Washington Post, New Scientist, MRS, IEEE Spectrum, etc. He is also interested in visual communication in science and engineering, and his essay on how to prepare scientific figures was the most downloaded article in Advanced Materials during the fall of 2011, more than 10,000 times. He received a 3M on Tenured Faculty Award, an SF Career Award, and was selected one of the TR35 GI by the MIT Technology Review. The floor is yours, Marco.
Thank you, Mehmet. Thank you so much for the invitation. It's always great to talk to the audience for the Terasaki Institute, and it was great to see you a couple of weeks ago, right, when I visited. Hopefully we'll get back to seminars in person soon. So today I'm going to tell you a little bit of a story really about how our group started looking into biological processes and ways to affect them, really, when I affect them, sort of controlling them using bioelectronic devices. And our inspiration really came from nature. Now we think about control, we always think about, I don't know, self-driving cars or, like robotaxis or drones, all these things that basically have some form of autonomy where, let's say, if a self-driving car veers off the road, there's a control system that kind of steers it a little bit one way or the other. But really, our bodies, nature is the champion of closed loop control. We just call it something different. When you think about natural systems or physiology, there's a phenomenon called hemostasis, which is basically like a way to maintain equilibrium. And the way it works is that, from sort of like a very conceptual way, there's something that needs to stay at equilibrium. And I'll show some examples. A typical example is temperature, for example. There's something that has to measure that particular variable that needs to stay at equilibrium. And if what's sensed is different from what variable is supposed to be, there's a control center that makes a decision and uses something called an effector in order to bring back hemostasis, right? When these things don't work, that's when we get sick, for example. Very, very, we all know, unfortunately, when we have, body temperature is really important. When it goes off one way or the other, it creates many problems. Sometimes it goes off intentionally when we get very warm, for example, when we try to defeat a disease. Other times it doesn't. There are many, it's a very complicated system, but the bottom line is that made of many parts, but the bottom line is that if our body temperature falls, what happens is that we sweat less, and our blood vessels contract in order to minimize the dissipation of heat from our extremities. And if it goes really bad, then we start shivering, which is basically an involuntary movement that makes us move really fast, and that creates heat, okay? On the other hand, if the body temperature goes too high, then, we sweat a lot, and then we have all this water that evaporates from our skin and it drops body temperature. Now, these things might sometimes overshoot, where for example, if you sweat a lot and then all of a sudden there is a breeze, you actually get really cold and you start shivering. And so these oscillations are typical oscillations of with any control system, where if you control things too fast, you get a big oscillation. If you control things slowly, then it takes you longer to arrive to the proper set point. This is what's typically called negative feedback, in that what happens is that you are bringing back, if something goes high, you bring it lower and the other way around, okay? Now, a very, very important, there's pretty much anything in our bodies works as some sort of equilibrium homeostasis, and the equilibrium is maintained by many parts. Something that is one of the very important ones is the sort of glucose insulin cycle, where we all know that insulin participates in absorbing or storing glucose in our muscles or storing in our adipose cells, and that we need to maintain a certain blood glucose in our blood in order to be able to function appropriately. If the glucose drops too low, we have problems. If it's too high, we have other problems. When people have diabetes, what happens is that our pancreas does not produce enough insulin or might not produce it at the right time. And so this particular cycle doesn't work. And we all know that one way to do it is actually to inject insulin at predetermined amount of predetermined times of the day in order to be able to bring this balance back. Now, doing this is difficult and it's time consuming. Therefore, one of the things that people have developed, this is work by Bailey and co-workers, was published in 2019 in Kidney International, is actually closed loop control. And the idea is quite simple. In this case, they have an insulin pump that delivers insulin to the body. The pump is connected to a glucose sensor. And when the glucose goes too high or too low, actually when glucose goes too high, it delivers just the right amount of insulin in order to bring it down. And you see what happens is that, like, if you have the closed loop control, which is the red line, maintains on average the glucose level lower, but really what matters is the envelope. So this envelope means the uncertainty of how much it oscillates. And you see that the red one is much better, okay? And this is what's called a closed loop control. However, in order to avoid all of these sort of like, big oscillations or issue with how much you deliver, there are, as part of electrical and computer engineering, as a matter of fact, there's an entire discipline called control that revolves around creating mathematical models and control system to make sure that equilibrium is maintained appropriately. And once I actually moved to electrical and computer engineering department about 7 years ago, and that's when I started thinking more about this issue of control in biological systems and how can we work with it. Now, about 10 years ago now, people started thinking of a different way to sort of like reestablish equilibrium homeostasis. Fundamentally, the idea is that, like, if we look at this situation for diabetes, the pancreas malfunctions, and so we need to provide an additional source of insulin in order to be able to maintain the appropriate glucose level, right? This paper that was written by Christopher Pham, Brian Litt, and Tracy Ed Boyden and Monsef Slahoui, this is like a position paper where their idea is that, like, well, why don't we think about making the sort of like targeting the pancreas, in this case would be, and making sure the pancreas does produce more insulin, even if it doesn't want to. And their idea is that, like, every time there is a malfunctioning, we look at biochemicals. Either we produce the substance that the malfunctioning organ is not producing, or we try to find a kind of like a biochemical cure to it to tweak the control. However, all of our organs, everything in our body ultimately functions or does actions after some form of neural stimulus from our brain through the nerves. And so what they say is that, like, well, what if we actually deliver impulses to the nerve that connects, in this case will be the pancreas, but it can be other nerves. And actually the vagus nerve is the one they usually target. And what if we were able to target the specific neurons in the circuit, and we basically tell the pancreas to sort of like produce insulin exactly when we want to, when we actually detect the glucose is too high. Basically, the idea would be to sort of like insert ourselves into the hemostasis using electrical impulses and to regulate the hemostasis. In this case, would be glucose, but it could be any other process, in a way that we bring it back to normal. And we then effectively either treat the disease or at least manage it in a way that the disease doesn't appear anymore. Now, they say it's electric. I would argue that it's actually ionic. And that, like, if we look at the way nerves propagate electrical impulses and signal, it's true we call it electrical impulses, but the bottom line is that these impulses come from the movement of ions. We are made mostly from water. Water is a very poor electronic conductors when it comes to moving electrons. However, we all know that salty water is actually a very good conductor of electricity. by means of ions diffusing or drifting in the water itself. And so our nerves, the way the action potential propagates in our nerves is actually through fluxes of sodium and potassium that move across the cell membrane, change the cell membrane potential, and it propagates like a soliton. In this case, will be from left to right. Therefore, what we started thinking about, if we really want to affect these kind of processes, it's true they're electrical, but the charge carriers, or if you will, the language that the body speaks are actually ions. Shouldn't we start thinking of ways to actually move and control the current of ions rather than just electrons? And so what we started doing in my group is looking at how can we use electrical or electronic devices or bioelectronic devices to move ions. And we started with the smallest one. We did a lot of work actually on protonic transistors in 2012. And then we moved to controlling the ionic concentration in solution. This is a schematic from work that was done in 2014. And what we do here, basically, we have a system that uses palladium metal. And this metal can absorb or push out H+ ions depending on the potential difference applied between the palladium metal and the solution. And so basically what we can do is using a voltage, in this case, V here, we can actually control the H+ concentration of the solution, which in turn is the pH. And we started with H+ because it was easier for us to do, but it's also important to know that pH actually tends to affect many biological processes. So this is an aside, like when we looked at the ion potential, the things that actually create the action potential are ion channels. So one of the things we started doing, we started integrating these PDH contacts with different ion channels. In this case, this is gramicidin. This is alamethocin that is gated depending on potential. And this is an example of a device in which we can apply potential here, V between the solution and the contact. There is a lipid bilayer that sort of like simulates the cell membrane between the contact of the solution. And depending on the voltage applied, we can measure the current across these ion channels, so we can make them open and closed. And we have published in this area as well. One of the first examples that we did in order to control, what happened here? To actually control, hold on, let me just fix this. Some kind of reason we have two titles in this slide. Back to normal. What we show here is that that was our first example of controlling biological processes using bioelectronics. So this is not closed loop, of course. What we did here, we actually used a simple system. It's called the luciferin oxyluciferin system. There's an enzyme called firefly luciferase that takes luciferin, makes it into oxyluciferin. The luciferin, as you can see here in the image, is not bright, and the oxyluciferin is actually fluorescent. Actually, is and this is actually a toy for kids, a Japanese toy that our postdoc Tako Miyake, who's now a professor at Waseda University, came up with. And the point being is that like this luminescent, not fluorescent. The point being is that like this reaction happens much faster at high pH. So here what we did is if normal pH, the reaction is slow, so we don't see much luminescence. And when we increase the pH by basically decreasing the H+ concentration in the solution by moving the H+ from the solution into this contact with the voltage control, we then increase the amount of luminescence in the system. Now this is what we've shown here is basically like a simple contact attached to a solution, but we wanted to do something a little bit more sophisticated so we can control. First of all, we can control bidirectionally. We can also control more H+ ions moving in and out. And we basically took a sort of part of a playbook that, these are ion pumps, and these are ion pumps were developed originally by the group of Magnus Berger in Sweden at Linköping University. And what we did, basically, we took a similar approach, but we still use our palladium hydride contact to basically incorporate the H+ into a solution. In this case, there's on the right here, there's a reservoir that contains concentrated HCl, about 1 molar. And there's this polymer here that is designed to filter out in such a way that only H+, only positive charges move from the right to the left when you apply potential. And then there is this PEI, this orange here, is designed to actually block the H+ from leaking, because obviously there's a very strong concentration difference between the right and the left, unless you apply voltage. And then there is a palladium here that absorbs the H+ with the proper voltage, and then it pushes out into a solution. It says buffer here because our big sort of achievement with this work that was published in APL Material, first author was Xenophon Strakosas, who's coincidentally now a postdoc with Magnus Berggren, was to be able to change the pH in the buffer solution. Now, most biological systems in our body, most processes are extremely sensitive to pH. Therefore, our body does not like big swings in pH, and it created a sort of defense mechanism against it, which is basically we are a buffer, both sort of like passively using through the carbonate system and actively through ion channels. So what that means is that we have a strong buffer capacity, and therefore we need, if you want to change the pH, we need to throw a lot of H+ at it, which is what this ion pump is able to do. Now, this is an example actually with the work we've done with Ali and Suryan, and there's a collaboration with Ali, Ada, Suryan, and myself, was funded by the Air Force. And what we've shown is that we can actually use this trigger of pH to actually deliver a cargo. So Ada is a professor at UCSD in pharmacology, and expertise is creating these particles that you see here in panel B. They are made by dextran, and what they do, depending on the pH, they will fall apart. So they're particles that can contain a drug to be delivered. In this case, we actually just have FDA, which is a fluorescent molecule that we use as a sort of like a proof of concept study. And these are designed typically for cancer therapy. It turns out that cancer has low pH because cancer cells reproduce really fast, and they sort of burn a lot of glucose and they produce a lot of CO2 in the process. So it's well known that localized cancer areas usually have a slightly lower pH. And so Ada has developed these particles to basically deliver localized chemotherapy, which is like a word so expert in this. But what we did, we basically showed that we can use our device with electronic control, by electronic control pH, to then make these dextran particles fall apart. These contain fluorescein that, when it goes into the cell, actually and then becomes fluorescent. And so we were able to show that we can deliver cargo to cells using a bioelectronic signal. All right. So a few years go by, we have these interesting devices, and we're like, well, what do we do with it, right? I started off talking about hemostasis and equilibrium. And so we started thinking about what are the kind of systems that we can control? And we started, I was lucky that a new colleague arrived in 2017. Her name is Marcela Gomez. She is now an associate professor in applied mathematics. She's an expert in control systems applied to synthetic biology. So we started looking at things we could do together, and we wrote this sort of perspective in cell systems. It came out in 2018. And the idea is what can we do in bioelectronics to really provide control to synthetic biology? Now, how did this work? Funded by DARPA Seedling, came this idea. What if we take a system of cells, right? Stem cells, they have infinite possibilities, but they don't know what they're going to be yet, and find out a way to sort of figure out, send different type of cells or the same cells in different areas into a different fate. The idea being is that when you have to create an organ, right? Not every part of the organ or tissue has the same cell. So you require some spatial temporal control of the final product. And so if you have a cell system like this, we're able to sense which state is it in, and then we can measure in space and time the expression of the different cells. We compare it to what we would like to be. In this case, let's say the green one could be like skin cells, and the red ones could be muscle cells, or like fat cells, for example. And then once we compare this, we look at our sort of control system, and the control system says, well, yes, you're going towards the desired state, or no, you're veering off from where you want to be. Therefore, we need to figure out a bioelectronic input to bring it back to the desired state. Fundamentally, is the strategy is a way to design ad hoc with spatial temporal control, a co-culture of multiple cells where each cell, you know exactly what's going to be, okay? So we started thinking about this, like, this is a really difficult problem. And so my colleague, Marcela Gomez, developed algorithms that are able to do this, and she employed machine learning. No, an extra machine learning, but we're able to write this review of perspective on how to use machine learning to control bioelectronic devices. This was in APL Materials in 2020. And the idea is, if you look at it, it's very similar to hemostasis in that, like, in this case, we have a sensor that senses the state of the system. We have a brain, right, which is the machine learning control that decides what to do with the state of the system. Then there's an actuator that sends the inputs from the machine learning control system into the biology. And then and then there's like the sensor measures again, and it's a cycle. Okay, so it's closed loop. This is where the loop gets closed. Okay. Now we decided we needed like a system to work with, and I was lucky enough to actually receive an email on Christmas Day of, I think, 2018. I still remember it because it was email on Christmas morning. And I'm like, who sends emails on Christmas morning more often? Who checks his email on Christmas morning? That was me. And it was an email from Michael Levin. Michael is a professor of biology at Tufts, and is famous for looking at bioelectricity and in regeneration. And he does a lot of really cool work at tissue regeneration. And his email was like, oh, I read one of your review on bioelectronics. Do you want to work together? And so I was like, yeah, that sounds great. Let's do it. And I have a great colleague to work with, Marcela, who's like the control expert. And so what should we do? Now, what Michael has shown is that cells in our body have different VMEM. Now, VMEM is the potential difference between the inside and the outside of the cell, which is, of course, an hemostatic process, hemostasis process, in which these ion channels move sodium, potassium, and different ions in and out of the cells to maintain this VMEM. VMEM is usually about minus 70 millivolts. However, and has many, many processes associated with it. However, what's interesting is that, like, all of these VMEM actually end up controlling different type of biological processes. And Michael calls this sort of bioelectric code of life. And actually, I think if you ever invite him, that would be a great person to invite to this series, because he has some really amazing things to discuss, and he's obviously can do that much better than me. Now, one of the things that he's shown, if he takes a planaria worm, what he's shown is that, like, he can actually, by hyperpolarizing or depolarizing the membrane voltage of the cells, it can actually program whether a headless planaria worm, after it gets cut, it regenerates. This is one of the things regenerates. Can have a head. If it's wild type, it's left like this. If it's both sides are depolarized, it becomes with two heads, or if once both sides are hyperpolarized, it comes with two tails. And so it seems that, like, in this case, the polarization of the cell membrane directly controls what the cell is going to do and how they're going to assemble to create one or two heads in this particular worm. Now, we didn't necessarily want to do this with worms. What we wanted to do is see if we can take a stem cell, which starts with very, very low membrane potential, and dial in a specific membrane potential here, and see whether by dialing in a specific membrane potential, we get something that is cornea, muscles, or fat. And then we were looking at muscle and fat. And the idea is that, like, you can do the bioelectronic experiment and then, like, do the sort of look at the genetic code of the cells and figure out what happened. Now, we didn't get to the, sorry, my kids just got home, so I need to make sure it's not too loud. We didn't get to a lofty goal of actually differentiating cells, but what we're able to do, we're actually able to at least show that we have closely controlled the membrane voltage for periods of up to 12 hours, which is the first step towards achieving the lofty goal of actually dialing in cell differentiation using bioelectronics. And this was published on the cover of Advanced Intelligent Systems in December 2020. So how do we do this? This is how it works. we decided to use pH as our first variable because it was relatively easy for us to do, and because there is a lot of the Michael work showed that basically, like, if you have a pH change, you can actually change the potential of the VMEM. What happens is that we have a bioelectronic device that can change pH. We have cells in the in vitro device. We change the pH. The change in pH will affect the voltage, the membrane voltage. We have a fluorescent transporter that is actually engineered in the cell, and the fluorescence of this particular dye goes up when the VMEM is high and goes down when the VMEM is low. We read the fluorescence, which in turn indicates the reading of VMEM using a fluorescent microscope. The images are collected, fed into a neural net that was trained previously that has an input target value that we decide, and then the net decides whether we want to actuate the bioelectronic device more or less, meaning that do we increase the pH or decrease the pH depending on the change in membrane voltage. Now, the first part is the device itself. This is basically a version of the device I show you made by Stracosa and co-workers. In this case, there's a reservoir here on the left. You can see the different materials are differently color coded. There is the palladium hydride and the silver chloride serves a reference. Then we have the yellow is the polymer that serves as a filter. The device is entirely made with Parylene and SU8. And in this case, this was a device that like microfluidic tape to seal it. As you can see in part B, there's lots of photolithography because at the time we were actually trying to also achieve high spatial resolution. So these are 100 microns each, and you see there's like 9 electrodes that are individually addressable. And this works if you have VH plus is positive, you decrease the pH of the system. If VH plus is negative, you increase the pH of the system. And the main difference from the Stracosas is that one just had a little droplet. This one has a target that contains cells. These are human-derived stem cells. And this here is actually PDMS, and it's a cap because you cannot, grow the cells on the device because the device is not going to be happy for like 24 hours, 1 or 2 weeks in a cell culture situation where there's like lots of water, and these are still electronics that they're waterproof, but they're not like they're not gonna be happy being underwater for 2 weeks. And so we grow the cells on a PMS cap separately. Then we just take the cap, put it in the device, and run the system. Now, one thing that you ought to know is that bioelectronic devices, they're not all the same. What that means is that you cannot say, okay, I want the pH to be, 6.5 in this area. You cannot just decide to give it a current of H plus and hope that the pH arrives at that area, because your input into the device is a voltage, but every device is likely different resistance and different efficiency. That means if you say apply 1 volt for 10 seconds to 10 devices, you're probably going to achieve 10 different pH changes. Now, that is a challenge because when you want to control the process, what you need to do is have a fine control of your input as well. So the first thing that we had to do in collaboration with Marcela Gomez was actually to devise a system that helps us put the pH in the right target. Now, in this case, the fluorescence here that you read is not the fluorescence of the cells. What it is the fluorescence of a pH dye that we put in solution. So there's no cells here. And what we show is that we can actually change the pH following a specific route. So what's plotted here is as follows. Now, the dotted black line is the target value that we program into the system. The red line is the actual value that we program into the system. The blue line here that refers to the y-axis on the right is actually the voltage H plus. Now let's just look at the one panel B here. So remember, in order to increase pH, you apply a negative voltage. to the device, and in order to decrease pH, you apply positive voltage to the device. Okay. Now, in this case, what happens is that like we're looking at fluorescence, which calibrates into pH. Now, the way this works is that like here, the fluorescence wants to go down. Now, the way this dye works is that lower fluorescence corresponds to lower pH, more acidic. So in order to bring the fluorescence signal down, which is this red trace here, we have to apply positive voltage for a certain amount of time. In this case, it's 10 minutes. And then when we want to bring it up, we have to do exactly the opposite, okay? And this is done by like a neural net that learns itself, which is important because every device is a little bit different. So you cannot just give it a calibration. And here, so we can try, we can follow pretty well, like this is just like a seesaw type of shape. This is like a sinusoidal function. This is a step function. And what you've seen here in F and G is that we can control 2 pixels. One here shown here on the E panel on the left, one on the right, sort of independently. That was important because if you want to sort of like give spatial resolution to this kind of system, you want to make sure that you can control independently in the properties of each area. Now, with that kind of like toolbox in hand, we take these cells, we set up a target intensity for the fluorescent voltage that basically corresponds to a specific VMEM. What we've learned is that the cells actually get really stressed out if you have the pH change constant throughout time. So what we learn how to do is actually that we had to float the device for a little bit, give it a little kick, then float it again, give it a kick, so on and so forth. Otherwise, the cells will get stressed out and die or just stop responding, as a matter of fact. Now here, what you see here, the fluorescence now is not the fluorescence of the associated with the pH, is actually the fluorescence of the fluorescent reporter that is part of the cell that reports the VMEM. Same trick as before. Here, the sort of the dotted line is the target value, the red line is the actual value, and the blue line is the H plus, the voltage applied to the device. You can see here. So this is what you would call a control in which the feedback is too fast, right? In that like it kicks too much one way or the other, and then like it kind of oscillates. In that like here we decrease the pH by increasing the amount of H plus ion in solution, which in turn decreases the pH with the positive voltage. That decreases, and that like makes the VMEM go, the fluorescent intensity go down, which means that the VMEM is going up. However, it overshoots, and then like, it was supposed to only come back here to the black line, and instead it overshoots. So now we have to bring it back on, so on and so forth. You can see these oscillations go down a little bit eventually. This is set up better when we try to maintain a certain voltage, a certain VMEM, it stays pretty well. Now, this was our biggest success, if you will. I know it's like a red line and a blue line, but it took a long time to actually do this. And it's important. One of the many pains we had is actually to make sure the cells will stay alive happily in the incubator and the bioelectronic device. And it took a fair amount of integration effort. Now, what we have here on the top is the fluorescence intensity, which corresponds to VMEM. And this on the x-axis is time, which we did it for 10 hours. The black lines are basically targets, intermediate targets. And our goal was to increase fluorescence intensity, which I believe means depolarizing the cell. And it took us some time, but we achieved it. And I think if I remember correctly, this was equivalent to changing the membrane voltage for about 10 to 20 millivolts, which is quite a bit because the bigger membrane voltage is about 70 millivolts. So we basically achieve a controlling VMEM cells for an extended amount of time. Now, in order for this to be useful to control the stem cell fate, this needs to happen for about 24 to 30 hours, and we are working on it right now. But the seedling that we had with DARPA finished, and so we had to start working on something a little bit different, which I will tell you about in a minute. Now, among other things, pH is very useful, but there's many other ions. So one of the things that we developed as part of this DARPA program, this was done by my PhD student, now Dr. Mampiangia, is looking at how to move other ions. And following the same trick that we did with palladium, where you have palladium, hydrogen, they can transfer from a palladium depending on the amount of voltage you apply. We basically decided to use, and palladium hydride is actually a reversible hydrogen electrode. So we decided to use that. Like, wow, why don't we use the most famous reference electrode, which is silver chloride? Now we always think of silver chloride as a way to sort of like have a reference voltage when we do electrochemistry. But the reason why it's a reference voltage is because the chloride concentration around the silver is actually maintained constant. And the way the electrode works, actually, there's an equilibrium in which chloride goes in and out of the electrode. Now, one of the interesting things, like we were the first people to actually characterize the current voltage characteristics of chloride moving in and out of silver in electrochemistry, because everybody always look at the voltage, but we were interested in what happens to the chloride ion. So our idea was that, well, if we have a positive potential on the silver, we should imbalance the equilibrium between the silver chloride and the chloride in the chlorine ion in the solution. So the chlorine ion should go from the solution into the contact, therefore reducing the chloride concentration in solution. Same thing. If we have negative voltage here, then we should push the chloride out, okay, and into the solution. And in order to do this, we use another fluorescent dye, MQAE. So MQAE intensity is high when there's a lot of chlorine ion in solution, and is low when there's not as much chlorine ion in solution. What we've shown here, very simple, is that when we apply 0.4 volts to the sort of like, and the nice thing about MQAE is actually very easy to calibrate as well. So we have 0.4 volts to the system here, plus 0.4. At 0 seconds, there's about 50. We start off with about 50 millimole. We apply 0.4 volts for about 100 seconds. Remember, the chlorine ion is supposed to go from a solution into the contact, therefore reducing the concentration. And yes, we can reduce it to about 30 millimolar as expected. Now, this was exciting. This actually is the first demonstration of chloride ion concentration control using bioelectronics. And again, the reason why this is important is because now you have all of these ways of using AI and control system to control the environment in which the biological system in the cells live and behave, and therefore we have a way to control them. Same thing when we apply a negative voltage, we're supposed to push chloride out, and we start off from now a relatively low chloride concentration of actually 0, and we can increase it all the way up to 50 millimole with about 100 seconds. Now we do the same trick using, we do a proof of concept on cells, same pump as shown before. Now the main difference is this. Now you see that polymer used to be shown in yellow here on the top left, panel A, the polymer between the reservoir and the target. Now is shown in green because the polymer in yellow was a filter that let only H+ go through. The polymer in green, on the other hand, is actually a filter that only lets chloride go through. Because otherwise, when you apply potential here, you will have things go from a negative to, you will have negative ions go from a negative to the positive, but you also have positive ions going from the positive to the negative, which is something that you want to avoid. Same type of cells, and you see that the layout is similar. There's a reservoir here with a big electron. There's a target here, and we actually we showed that this is on C is the fluorescence of MQAE, which indicates the concentration of chloride. And on the right is actually the fluorescence of arc light. Arc light is the reporter that indicates VMEM. What's shown here in E is that when you give chloride, you actually go from a high VMEM to a low VMEM. So we're able to, we have two ways to control chloride VMEM, either with H+ or chlorine ion, and now we work, putting everything together. Now, as I said, what happened, the acetylene turned into a different project. And as part of this work, we were awarded a grant as part of Bioelectronic Controller Wound Regeneration Program called BETR by DARPA. And we're currently working on it. And hopefully if I get invited back in 2-3 years, I will have all of the results of these bioelectronic components applied to wound. The idea is the same. Basically, we have bioelectronics that deliver specific substances, ions and small molecules to the wound, depending after measuring the wound state. And we do so using different type of reporters. And this is like I'm lucky to be able to lead a really great team of people. Marcella, who's worked with me before on the control loop system. Misha Tedorescu, who came in as sort of like a device integrator, because now we have like bigger device with lots of electronics. Michael Levin, who's works with us from Tufts University, and Dr. Issaroff and Dr. Min Zhao. Dr. Issaroff is the chief of dermatology at the Veterans Affairs in Hospital in Davis, and a professor at dermatology UC Davis. And Professor Zhao is also a professor of dermatology at Davis. So we have some exciting work happening that I cannot share quite yet, but I'm putting a plug there because there should be some good stuff coming out soon. Now, with that, I've talked about for about 40 minutes, I think, which is probably a reasonable amount of time via Zoom. I would like to thank my group. This is actually a little bit of a sort of like a vintage picture, if you will, but Jack Selberg, Dr. Selberg, actually just brother, was one of the sort of like the people doing most of the work on the devices, and really was the architect of all of these devices. Dr. Gia is also graduated. She did a lot of work for the ions, and Arika is a 4th-year PhD student working with me. Now Nevio actually went to work for Neuralink, and Kerry works at Ford, and Brian is still working with us. And with that, thank all of our collaborators, thank funding, mostly from DARPA for the recent work, and special thanks to program manager, Paul, who's been really instrumental as far as directing this type of work. Thank you for your attention. I'll be happy to answer any questions.
Thank you for this exciting talk, Marco. I appreciate it. Couple of things. For the changes in pH, what resolution do you need for your sensor? Do they have to be nanowire sensors, or?
So the one thing that I've learned is that, like, if you try to actuate something using electronics, it's much easier if you actually like sense it with a different system. And so the way we sense it, we actually use fluorescent dyes. So these are from Fisher Scientific, they're dyes that you put in a solution and they change fluorescence depending on pH. So we put them in our solution and we do all this work under fluorescent microscope. So the loop is basically a fluorescent sensor. It's an optical sensor, right? Fluorescent sensor. The system reads the fluorescence from the image and then decides what to do. And that gives us pretty good spatial resolution, as good as the microscope, which is, a micron or 10 microns or something like that.
Thank you. Regarding the cells, would your system work with single cells, or how many number of cells do you need?
Yeah. There's nothing that stops it from working with single cells. One of the things is, it requires a little bit more sophisticated lithography. Right now, our pads are about 100 microns, so you would need to do it with a pad that's maybe 100 microns, maybe a little bit less, right? Single cell is like 40, 50 microns. The other challenge is that sometimes these cells die, right? So if you have lots of cells, a few dead ones, it's not a big problem. If you have one cell and dies, then your experiment goes belly up. So
I see. One other question. A few years ago, there was an article in Scientific American about bioelectronics for pharmaceuticals. I think Big Pharma is really interested in this area. What are your comments?
Oh, I don't know. I think there's a lot of really nice work happening there. It's really, I think I showed the Nature perspective, but Scientific American was the same thing. And I think there actually are treatments already in the clinic. So it's really an exciting way of doing things. And in a way, this work, it just looks more at the interface using ions, just a different way. But I think it's really exciting that there's different ways of treating people. The more, the better, right? Because obviously we all know that we need more treatments.
Well, that's all the questions I have. Thank you for your time, Marco. I truly appreciate it.
All right. Thank you so much.
Thank you.